Thanks for the introduction and thanks a lot for the invitation and for the organization of this course. So my name is Georg. I'm from Medical University Vienna, and I'll be talking about matching and studying multivariate patterns across individuals and go a bit more into detail about what we can do in order to deal with complex patterns and how to study them across individuals, even if there are kind of substantial differences across populations. And I put all the slides and also the links to the relevant papers I will be mentioning during the talk on this URL. So it will stay on the bottom of the slide. So if you can go there, and there will be also links to the code that's necessary in order to play with these methods. So the rough agenda is, I'll talk very briefly about normalization, but I think most of you will be very familiar with what people do and what you do when you are working with population level fMRI and MRI data. Then I'd like to discuss a bit about variability, about different aspects of variability, and also about insights we get from studying the discrepancies of this variability across individuals, like what's different in terms of structure, what's different in terms of functional architecture, and how we can kind of cope with the discrepancies and the possible decoupling of those two. And then I'll mention a couple of useful approaches in order to deal with that and in order to do MVPA across large cohorts or across very different cohorts. But I'll go into detail only for two approaches, and one of them will be dealing with response patterns. So how can we map response patterns? How can we find matching response patterns across individuals? It will be hyperalignment, introduced by Jim Haxby and his colleagues. And the second one will be, what if we don't want to use task data, because maybe we want to use task data for the subsequent analysis, but what if we want to use some neutral basis for finding associations across individuals? And that will be, in this case, an example using intrinsic connectivity patterns. And then all through the talk, actually, I'll show some results and examples in order to illustrate what it actually means. So I think all of you are familiar with normalization. Everybody's doing it when you're studying any phenomena in a population. And the typical approach to normalization is to use the anatomical landscape, the anatomical structure of each individual in order to match corresponding points across persons. And the underlying assumption here is that we will do a pretty good job in matching homologous, like corresponding functional regions, once we get the matching of the anatomy right. And that is actually quite successful, so we can do very nice population studies with that, but it has a couple of limitations. So one of them is that we can only ask a certain subset of possible questions. So we can talk about differences across subjects, but we are forced to mix together the differences in the location of function and also the difference in the actual structure of function. The second one, and that's particularly relevant if you are working with clinical populations, is that there is always the danger of confounds. So there are diseases, there are, let's say, lesions, tumors that do affect both the function and the structure. And then if you're only able to analyze both at the same time, then you might not be able to discern if the tumor has just shifted aside a functional region or really kind of impacted the functioning of this region. And so what I'd like to discuss in the next couple of minutes before we go to the methods is this kind of felt discrepancy between anatomy and function, and how the variability across individuals might be better viewed as a combination of those two, and why effort is worth taking to kind of discerning that. And so one example, which I think is kind of just an illustration. So these are two experiments with HCP data. So just taking a couple of subjects, doing activation detection with a standard GLM on each of them, one for motor and one for language. And the colors here show basically just the overlap of the detected regions across these subjects. And you see that for the motor regions, we are doing pretty fine for some central spot where we get almost 100% of the people have the activation at the same point, whereas for language, that's a far lower threshold. So like at the best case, we're just getting 60% overlap between, or 60% of the persons have an activation at the same point. And so there is some extensive work in this variability, or some people call it individuality, of the response patterns and connectivity patterns across people. So this is one paper by Sophia Müller, and she studied how functional connectivity varies across individuals. And the interesting finding was not so much the fact that it does vary, but that it varies very differently on different portions of the cortex. And so if you look at this map where basically blue regions show regions where there is very little variability, and red regions and orange regions show regions where there is a lot of variability across individuals, you see that more like the primary regions like motor and vision have very little variability, and the other regions have a lot of variability. And when you kind of think back to the last slide about how consistent are functional activation localization results across individuals, you start to think about whether part of the reason for this functional variability across individuals is the actual variability of the functional architecture in these individuals, but part of it might be just that the very same functional connectivity architecture is just sitting at slightly different locations. And so when you are talking about that, you should be aware that there are these two components of variability. There's two very, I think, worthwhile papers to read, one by Sepulcre and his colleagues, who looked at the structure of these connectivity patterns. And when I talk about connectivity, I always mean basically signal correlation patterns in resting state fMRI. So they looked at the spatial distance between connected areas. And so here, again, in the motor and the vision center, you see very kind of short-range connections, whereas in these other centers, you see very long-range connections. And their hypothesis was that in these areas that are blue here, we have a very kind of congealed, very hardwired, canonically short-range connectivity structure that kind of does the calculations job, whereas in the other orange regions, we have very wide-ranging networks. And so I think the takeaway message from this range of work is that we have differences across individuals. We can kind of split up. It's worthwhile to split this up into spatial and functional differences. But I think the main thing that we have to start to think about if we are really looking at fine-grained patterns is that we need to find alternative ways of talking about correspondence across persons. So just saying I'm sitting on the same point on the cortical surface is not enough if you are studying multivariate response patterns or processing patterns in individuals, because you will not be at the same site in a different person if you're looking at the same function. And some interesting observations about that linked to a lot of insights in evolution and development are that this variability is not just something that's uniformly distributed on the cortex, but very, very different in different areas. And this difference between variability and areas, this connection, also relates to a lot of thoughts and a lot of kind of ongoing work people have into how the structure of our connectome came about and how brain develops overall. So here just a few examples. So this is a map of the very late cortical expansion between macaque monkeys and humans, and you can see there's a bit of a proxy about what are the most modern areas of the brain, and you see that here these orange areas are again the ones where we have very high variability, but these are also the ones where there was the most pronounced expansion in the very late stages of evolution. So these are very human-specific areas that are kind of dealing with very complex and very human-specific cognitive abilities. And I think it's interesting to have that in mind when we study these abilities, that these are also the regions where you have to think a bit more careful about what does correspondence mean and what kinds of correspondence we actually can rely on and what kind of notion of correspondence makes sense if you're talking about individuals. And this is fetal MRI data, so we can also do fMRI in the fetus in order to understand the very early emerging of these functional networks. And again, you see a very similar pattern here when you look at how different functional networks kind of emerge during this very early phase of development. And one interesting thing is when you look at the sequence of this emergence, we again see a very similar pattern that like occipital areas, vision, come first and the parietal and frontal areas come last. And so I think there is a lot of work still to be done in order to understand this relationship, but I think it's just meant as something to embed what we will be discussing during the rest of the talk. It means that when we talk about correspondence, that's not a trivial thing. It means that we have to think about different aspects here, and we should always be aware that this is linked to many, many different research questions from evolution and development, and ultimately a better understanding about how brain architecture emerges and how it kind of does the job it does. And one, of course, specifically difficult application here is when you're looking at clinical population, where the variance, the decoupling between the anatomical framework and the function might be very severe because there is just reorganization happening once parts of the brain are impacted by disease. All right, so the remaining talk, I'd like to talk about 2 ways to find correspondence between multivariate patterns and then to kind of take this and use it in order to do better studies of these multivariate patterns in order to get more specific results, but also how can we use this fact that there might be a decoupling between anatomy and function as kind of a marker, as a quantitative measure that tells us something about populations, that tells us something about the difference between different cohorts. And the 2 things I'd like to talk about is first, when we have task fMRI, can we map response patterns across individuals? And I'll be mostly talking about methods introduced by Jim Huxby and his colleagues. And the second one will be about how can we map connectivity patterns across individuals if we are relying on resting state connectivity, either because it's available or because it spans a much wider area of the cortex, or because we might like to use these registration results in order to be a basis of task-based analysis, and we don't want to confound the kind of means to do the registration with the actual object of our analysis. All right, so let's start with hyperalignment. There is some code online, but again, like all the links to the codes and the papers are on the webpage I put up for this talk. So the basic idea of hyperalignment is that if you have multivariate response patterns that are happening at different areas of the cortex, then one way is to align the anatomical structure and then believe or trust or hope that the same kind of components of these patterns will sit on the same side of the cortex. That's not always true, and that's kind of almost never true. And so Jim Huxby came up with the idea that we might do the alignment not in the native kind of anatomical space, but like let's have a look at the response patterns of individual voxels and let that be the feature that we use for the alignment, that we use for establishing correspondence across individuals. And so what you see here is that we have different responses to different stimuli, or just like a sequence of responses. If the subject is watching a movie, we have different subjects. And we can view these responses as points in a feature space. And when you know something about Procrustes alignment, you can basically then perform the alignment in this feature space. And so we have the original space, we have the align space, and then we can do some more processing in order to kind of break down the dimensionality of this space and get a bit more manageable dataset characteristics by, for example, just doing PCA. And this actually fairly simple pipeline allows us to achieve a representation of the stimuli of the individual response patterns that we can then use much better to transport something that we've learned from one subject to decoding from another subject. So the basic idea is that we have a new subject, we have a response pattern, we project it into this space, and then in this space, in the features that we get from this space, we can decoding and decoding if you're doing MVPA. And the results, so here just a brief summary, so we can do either the decoding in the native voxels, or we can do it in this joint space where the alignment has already happened, but the alignment has happened on the response vectors for each subject. So we can basically work in a representational space where we can ensure that similar response profiles are mapped to similar response profiles across individuals. And when you use this for decoding, you see something very nice, and that's, so the green bars are basically if we train a classifier on a list of stimuli and then try to decode the stimulus someone is looking at or someone is experiencing while lying in the scanner from the fMRI data of this individual subject. And if you basically trust the anatomical alignment, then you're getting an accuracy that's here shown with the green bars. But if you do this representational transformation of the response pattern and do the alignment in the response representational space, then you get a much higher accuracy. And that's very consistent actually with the accuracy that you would get if you would try to decode the stimulus pattern from the same person. So we are basically arriving at the accuracy of within-person repeatability, even though we are transforming or transferring codes and decoding patterns across individuals. And here they are just the confusion matrices. So these three confusion matrices are basically working in this joint embedding space. And this is a confusion matrix of the stimuli, basically showing how many of the classifications went right and how many of them went wrong if you just rely on the cortical function, structural registration for your analysis. And so when you think that a bit further, it gives you a very nice tool in order to correspond, in order to be basically a bit independent from accurate anatomical registrations. And that might be helpful if you do registration across individual persons, but it's even more helpful if you want to compare across species where you really don't have a very good idea about what correspondence means and what correspondence function means with respect to corresponding anatomy. And so this representation is, I think, very, very nice and very helpful. And so that's an idea that came up by for example, Nico Krieger's court. And the idea is that I don't want to look at individual voxels. I don't want to have to find corresponding voxels across individuals, but I just want to characterize the coding characteristics of a certain part of the brain. And I can do that by basically letting the person or the monkey or even the machine or deep learning algorithm or whatever you're interested in experience different stimuli and then look at how similar the response patterns are, or how easy it is for my classifier or for my decoder to differentiate between different stimuli looking at the response pattern. And then it turns out that for a group of stimuli that might be very easy, basically to get confused because they have so similar response patterns, and for other kind of segments of your population, of your stimulus population, it's very easy to differentiate between because the response patterns are very different. So here in this example, just like a blue point means that you have very similar response patterns, whereas a red point means you have very different response patterns. And so by this analysis, you are now able to, for example, compare the overall structure or the overall decoding characteristics, let's say on a very summary level, though, but still you are able to compare them, for example, between humans and monkeys. So these representations calculated from these two different species, or even if you want to compare this, if you have trained a deep learning architecture, you can compare these response patterns between humans or animals and machine, which I think is interesting in order to probe different parts of architectures that might be artificial, but you have now a tool at hand to actually see whether they are behaving in some way similar to natural architecture. And all of this is basically done by removing the necessity of relying on anatomical correspondence. At the same time, it of course limits your kind of specificity of description because you, after all, it's not really that you are looking at the complete patterns and compare them, but you're just looking at the response characteristics of these patterns in different individuals or different species. Okay. The second idea I'd like to go into a bit more detail is, right now we talked about specific response patterns, about specific stimuli and tasks and how to find them across individuals or species. But it might be handy for your specific problem to have some kind of neutral means of establishing correspondence so that you can say, well, this is the corresponding area, or this is the corresponding configuration of points in one person and another person, so that on top of that you can do individualized kind of task-based analysis. There are a couple of approaches, so two of them are shown here, so I think many of you will be familiar with Emma Robinson's work, which is, I think, by now also part of the releases of the HCP data, so she used multimodal data, not only task data, but also a lot of structural information in order to find correspondences across individuals and do that in a much better way than if you would just use the structural information. So this is this work. There is a very, I think, interesting paper by Tavor and his colleagues, which is about how can we use resting state and other multimodal information of brains of different individuals in order to predict stimulus responses. So they basically train the predictor on a subset of the population, given structure, connectivity, DDI information, resting state fMRI information, predict the responses to specific stimuli, and then if you try to use this predictor on a new subject, it works actually fairly well, and it can kind of capture the individual differences very well, which basically tells us that the differences that we capture with using DTI and fMRI and resting state fMRI and all other structural information has a much tighter kind of connection to task-based activations. And I'd like to go in a bit more detail on ways or means to use intrinsic functional connectivity in order to find corresponding regions across persons, because it's actually pretty analogous to hyperalignment, but just doesn't use any task information, but just uses resting state signal correlation patterns. So imagine you have a brain and you are able to measure resting state fMRI across time, across the entire brain. That gives you a lot of signals and also gives you a signal correlation matrix. And you can use this correlation matrix basically to create an embedding where every point in the brain is embedded in the embedding space at a specific position. And the way this embedding is designed, and I'll show you how it works in a second, is that areas that are very highly correlated or that exhibit very similar behavior are mapped to very close by regions in this embedding space. And in reality, these matrices look somehow like this. So it's a very big graph. Only part of these edges are actually used based on some criterion that you basically introduce in order to say, well, this level of correlation is reliable and this below this level it's not reliable. We can use an embedding approach, and I only go very briefly through that, but basically it means that you can use this covariance matrix in order to create an embedding in a closed form way. So you do just have to do a singular value decomposition of a reweighted matrix. And the nice thing is that the connectivity through the entire graph in your brain is then mapped into a Euclidean space where the Euclidean distance is informative. And this Euclidean distance becoming informative about the correlation pattern means that you have a structure that represents this functional connectivity pattern of the brain just in an embedding space, but now lets you basically apply everything you can do in a Euclidean space. In reality, this looks somehow like that for a brain. So we have points in the brain that are mapped into an embedding space. If you do clustering in the embedding space, you can color these point clouds, and you see that they also give kind of sensible clusters in the brain. But the really interesting thing is, or I think the really helpful thing is that when you do that from multiple individuals, it turns out that this embedding structure, this representation of the global functional connectivity architecture is stable enough so that you are able to use it in order to do registration. So similar to think about structural data, and brains look similar enough so that I can map the brain of one person into another person, these representations turn out to be stable enough so that we can, by just deforming one of these point clouds, rotation, scaling, and so on, translation, and a very slight nonlinear transformation, we can map them into another person's representation. And this gives us a means of mapping between positions across persons, even if these positions might actually be sitting at completely different areas on the cortex. Yeah, I leave that out. But I think the important thing for us, if we are studying multivariate patterns across cohorts, is that we can basically summarize this into one model and do that in a group-wise paradigm. So we can create one model of this point cloud and learn it based on evidence from multiple individuals. And that gives us, on the one hand, a population-level representation of this connectivity structure, and on the other hand, a means of transferring information from one subject to the other. And so there is very nice work by Daniel Margulis and his colleagues who did that on a very large population. And so this is the embedding result if you do that, I think, on about 1,000 subjects. And what's interesting here is that when you look at this, so each point here is one point on the cortex. This is the model that is generated from a population of 1,000 individuals. So if you just color these corners and map them back to the cortex, they give very sensible and very interesting region. And essentially, you are getting the transition from more unimodal to more heteromodal architecture parts in the brain represented in this embedding. So we are able to kind of collect a population-level model of functional connectivity from a large set of individuals. And where it gets interesting, if you want to do studies, is to go back and see where is this coming from in each individual subject that contributed to this joint representation. And so think about this distribution here. So that's the kind of population level average of mapping these coordinates back to the cortical space. And we can do the same for an individual. So we can basically look at each individual coordinate of a point and map the coefficient of this coordinate back to the point where this point was coming from the cortex. And if we do that across a couple of subjects, we notice that it largely looks the same. So this is the first component, second component, and so on. So they look largely the same, but still we get some individual differences. And now the hypothesis is that these differences, if we can kind of compensate for them, we get a better alignment and a better chance to study task and response patterns across individuals. And the idea here is, and that's a paper by, I was not involved, but also Paulina Golland and Satrajit Josh, was to use this information and use it as a cost function in order to do cortical alignment. And now when you try to test it, the first thing is, of course, if we base this alignment on these connectivity patterns and shift the cortex around so that these, I hope I can go back. So if that these profiles are better aligned, we make a better job. Or our hypothesis is that we make a better job. But the first sanity check, of course, is if we do that for the same person, how much of a difference will we get? So this kind of visualizes how much of a shifting around you get with registering across the same person of multiple scans. So it's very little, it's still there. There is some noise, but it's fairly minimal, especially if you compare it to the deformation fields you get when you do this registration across individuals. And so here the colors basically show in these red areas there's a lot of variability of the same connectivity profile sitting on different locations on the cortex, whereas on these regions there is a lot less of this variability. And this basically tells you that part of this variability we looked at before can be explained by just cortical shifts or shifts of the position of functional areas along the cortex. We can use that on one hand in order to improve alignment a little bit and get more specific response patterns if we then do task-based activation analysis. So this is one example when we do task-based analysis. I think it's a relational task in the HCP dataset, and we do alignment, perform alignment just on anatomical data, we get this result, and if we then add the resting state data, we get a bit more sharper, a bit more specific results in our task activation. And the reason for that is that we have compensated for slight position differences of these functional areas, and now we are really looking only at the differences in the internal, like functional structures that we are observing. So that's one thing. But the other thing, and that's why the talk is also called Studying Multivariate Correlation Patterns or Patterns, is that now that we have a tool to compensate for different locations on the cortex, we can also use this kind of decoupling as a feature of a cohort. So we can say something about whether a cohort is more variable, whether there is more decoupling or less decoupling in other cohorts. So this is one very basic example. So that is a paper by Karl-Heinz Nenning, where the goal was to see if we can get anything quantitative out of this. And one thing is that we can check the individuality or differences in the individuality and how tightly knit is the connection between anatomical location and function in one cohort and the other cohort. So these were just left and right-handed subjects. And we can also, since we can do a mapping from this joint space into the cortex spaces of individuals, we can look at the relative size of different functional areas. So we can say, well, this corresponding functional area or this group of vertices on the surface, it's rather large in this cohort and rather small in the other cohort. So it gives us not only a tool that's kind of a pre-processing pipeline part, but it also gives us a tool of actually doing analysis of cohorts and across subjects by allowing us to quantify the link between anatomy and function between persons. Where this is helpful, just from a practical point of view, is in clinical populations. So this is an example where there were tumor cohorts, a tumor patient, and when you use this mapping as opposed to just anatomical mapping. You get a much better kind of accuracy in reconstructing the area where, for example, language resides. You get a very kind of sensible mapping between left and right lateralized subjects, language lateralized subjects, because it's very straightforward to find these corresponding regions in these embedding spaces, whereas it's a lot less straightforward because on the anatomical space, because they might just be sitting at completely different parts of the brain. So basically it allows us to link this functional correspondence across subjects. And so that's my last, or one but last slide. So I think when we think about multivariate patterns and when we think about how to study them across subjects, either because we want to find a shared characteristic, we want to find common mechanisms or features of these patterns across persons, then we also should think about what correspondence means. And I think we have tools that can help us to establish correspondence. And I think establishing correspondence across patterns, either by hyperalignment or by other methods, can not only help us to do better group analysis by basically getting better correspondences, but is actually something that's informative because it tells us something about the relationship between anatomy and function, and ultimately about what of this is actually important and what of this might be variable because it just doesn't matter that much. And so these were just a little bit food for thought. So this is a map of how variable cortex parcellations are in different areas of the cortex. Here we again see the map of how differences in the location of functionally equivalent areas are distributed across the cortex. That's a map of the evolutionary expansion, so this expansion between macaque and humans, where basically we can see that the kind of how modern different areas are, very roughly speaking. And here is a map of how much benefit we gain from doing actual functional alignment as opposed to just structural alignment. And I think there is a lot of similarity. There is also a lot of things that are naturally quite different. But I think that beside that being a tool or a kind of a toolbox of approaches that are emerging right now that help us to do MVPA across subjects, even if there is heterogeneity in the population, it also is a means of starting to essentially know more about the relationships between why does the brain develop in the way it does, and what matters and what does not matter, and what is easy to reconfigure in the case of disease and what's not. And I think that was basically the summary. I'd like to, so you don't have to write it down all right now. So these are the papers most of this talk was based on. And again, so here is the URL. So all of the papers and all of the code by everybody who kind of did code in this direction is available on that website. And with that, thanks for the attention.
We have time for a couple of questions if you have questions. No question? Or you can hold on to them when we have a question session later on. So I think we can thank Georg again.